If we're going to talk about allele frequencies and how the frequencies of a particular trait change over time, we have to have a way to actually measure that. The way that we do this is with something called the Hardy-Weinberg equilibrium and the Hardy-Weinberg equation. So Hardy and Weinberg were two independent scientists who developed this equation to determine how frequent the genotypes and therefore the alleles are in a particular population at a particular time. So the Hardy-Weinberg equilibrium is a set of sort of conditions that say that when a population is not experiencing any of these conditions, they are not evolving. But it can also be used to calculate the allele frequencies in a population. And if you calculate the allele frequencies before a suspected evolutionary pressure and after a suspected evolutionary pressure, you can actually see if they have changed. So the Hardy-Weinberg equilibrium and equation are important because they allow you to observe whether or not allele frequencies are changing and what factor might be causing evolution. So the Hardy-Weinberg equilibrium actually describes a hypothetical population that is not evolving. In reality, all populations are experiencing some sort of evolution all the time. Now this might be a slight change due to chance from year to year, or it might be an actual pressure such as global warming or deforestation, things like that. And in a real population, we see that a variety of these conditions are never going to be met, or at least they're never all going to be met at once. So basically, we never have a population that's in Hardy-Weinberg equilibrium, but you can sort of assume that at a specific point in time, when nothing is going on today, that population is in Hardy-Weinberg equilibrium. So it's sort of a cheat, but we actually do use it to determine at one point in time what the allele frequencies and genotype frequencies are, and then at another time what they are. So let's talk about what these Hardy-Weinberg conditions are. Now I'm going to do this video in two parts. There are 5 conditions. I'm trying to keep them a little bit smaller. So we're going to talk about the first 3 in this video and the next 2 in the next video. So these are also called Hardy-Weinberg assumptions, conditions, criteria, whatever you want to call them. These are characteristics of a population that we are going to assume are not happening, evolutionary pressures that are not happening at a particular time. Because if they're not happening, if none of these 5 things are occurring in a population, that population is not evolving at this point in time. Please keep in mind that the Hardy-Weinberg is talking about one trait at a time, or one gene at a time. So let's say you're looking at a bird and you might be interested in their beak size. At one particular point in time, their beak size might not be evolving. There might not be any pressure from the outside. There might not be any chance events that are changing the structure of the beak within the population. But maybe at the same time, those birds are being selected for their wing coloration. Just saying that one trait is not evolving does not mean that the entire population is not evolving somehow, because there's always probably some trait that is being selected for or acted upon by chance. So Hardy-Weinberg equation and equilibrium talk about one gene at a time. Is this one gene evolving in a population? So let's talk about these assumptions, keeping in mind that if any of these are violated at any time with respect to one gene, then your population is evolving. So our assumptions: no natural selection. That means nobody is surviving any better than anybody else. No new mutations. That means we're not getting any new alleles. No migration. That means no one's leaving a population or coming into a population and moving their alleles with them. Infinitely large population size. Or no genetic drift. There's kind of 2 different ways to say this one. I'll talk about what genetic drift is, but basically you have to have a population that is large enough that chance events are not going to affect the allele frequencies. And then our last is random mating. So nobody is being forced to inbreed due to small population sizes, and nobody is choosing your trait as a sexual selection, as something that they're choosing you for. They're not choosing you for this trait. So we're going to talk about 1 through 3 here in this video. So first one, no natural selection. This is usually the most obvious one. This means that individuals are displaying heritable genetic variation in a population. And we know that more offspring are produced than can survive. And this is because of competition for resources and predation and finding a mate. So we know what natural selection is at this point. Some individuals will be better able to survive and reproduce. This one is a hard one to meet. If you're wanting to know if a population is in Hardy-Weinberg equilibrium, natural selection is probably a pretty hard one to determine or to control for. So if we're violating this, if natural selection is occurring in a population, we have differential success, differential reproduction. We have variation in those heritable traits. So some individuals are going to survive better than others. And then, of course, this means that if the population is evolving, that trait is being passed on to the next generation. No mutation. So we just talked about mutation in the last video. We've talked about mutation several times. So again, a mutation is any change in the sequence that affects the phenotype. This one is important because, remember, mutations produce new variation. And if you produce a new allele, you decrease the relative frequency of the other alleles. Mutation is important because it introduces new alleles, which affects the frequency of other alleles in the population. Mutations are going to occur at random with respect to the individual. So this means that a mutation does not occur to make this individual survive. A mutation occurs and it's good luck or bad luck. Sorry. You have a mutation and it produces a new allele and you die, or you produces a new allele that doesn't affect your reproduction. But when we're talking about Hardy-Weinberg, we're assuming that no allele is coming up that is giving you a selective advantage. This doesn't mean no mutation is happening somewhere else in the genome. It just means that no mutation is happening in the allele that you're studying. This is usually a pretty easy one to meet because mutations are generally very rare. And so not only are they rare, but it's really unlikely that that rare event is going to happen in the allele that you're studying. So no mutation is a pretty easy condition to meet. Also, in a large population, mutations usually have negligible effects. So you probably won't really see much of a change in allele frequencies based on this mutation. At least not until that mutation becomes more prevalent. The other thing with mutations is you're not going to get multiple mutations in the same gene that you're looking at in a short amount of time. So we don't have to worry about mutation too much. Our third one is no migration or gene flow. So we all know what migration is. It's when an individual leaves where it is and moves somewhere else. This is important for evolution because when it leaves its population, it takes its allele with it, which means, or its two alleles with it. And so it means that it changes the frequency in the population that it left, and it changes the frequency of the population that it moves to. So this idea of gene flow is the movement of genes between two gene pools or two populations. So depending on your population, you might have individuals moving in, you might have individuals moving out, or you might have both. If your population is in Hardy-Weinberg equilibrium, you're not going to have gene flow. There's nobody coming in bringing their genes and nobody going out bringing their genes. And therefore, you don't get any changes in gene frequency because of that gene flow. Now, gene flow is actually quite important, and it's important for a population, and it's important for speciation. So gene flow actually helps maintain diversity in a population. So this goes back to sexual reproduction. If you have an individual from another population, it's bringing its variation in with it. And so then when you reproduce with that individual, you get new shuffling of alleles, and now you have more variation than was previously present. So this is actually a good thing, because if you have variation in a population, it allows that population to respond to pressures. So you're not completely wiped out if something changes, because there's probably some individual that has a combination of alleles that's really advantageous. This is also important because it reduces genetic differences between two populations. So if you are inbreeding with a population, it keeps those populations more similar. And this is going to limit speciation. If you can interbreed with another population, you not only maintain genetic diversity, but you also maintain one species. If you're isolated, if you're not exchanging genetic information for one reason or another, that's when you can start to get new species. Each population sort of takes its own evolutionary track, and eventually, if that track diverges enough, you're going to get new species. And of course, that's evolution. So if we're trying to meet this Hardy-Weinberg condition, no one's moving in and no one's moving out, and so you're not getting a change in allele frequencies because of that. So I have a couple examples of gene flow here, just to kind of explain what gene flow is. So if new alleles come in, they change the relative frequencies of all of the other alleles. Or if the same alleles come in, so it doesn't have to be a new allele, the same type of allele can come in, it still changes the frequency. And so the frequency can change based on the number of individuals that come in, how prevalent that allele is in the population. So this diagram is not from our book, it's from one of my genetics textbooks. But this shows the gradual movement of the B blood type. So this is showing the A, B, and O blood types. Well, actually, it's just showing the prevalence of the B blood type. So we know there are A, B, and O blood types. The B blood type actually came from the Mongolian people, and once the Mongolian people started to move out of Eastern Europe and into Western Europe, they brought that B allele with them. So you can see on this far right-hand side, the far eastern side, you have a high percentage of the population with the B allele frequency. And of course, those individuals are eventually going to interbreed with local populations, and they're going to spread out and move into those populations, even if they're not breeding, even if they are not encountering the local people too much, or I guess interbreeding is the right word. If they're not interbreeding too much, they are still bringing that allele into the population. As you move farther west, you can see that the percentage of the B allele decreases. So in some of these regions, you have less than 5% of the population with that B allele. So this is just showing how that allele can move over time and change the frequencies in a population as individuals carry their alleles with them. So this is an example from our book of gene flow. So gene flow is important because not only is it changing allele frequencies, but it can also affect how well a population is adapted to the environment that they're in. So gene flow can bring in advantageous alleles. It can also bring in alleles that are actually detrimental to the individual or the population, because if they're not adapted to the place that they're coming to, they're going to be at a disadvantage reproductively and as far as survival goes. So our book talks about this Dutch island called Vlieland, and the example is a songbird, this pretty blue and yellow songbird here. In this case, you have an island, and islands are, of course, good examples, good situations in order to study evolution because they're isolated. or mostly isolated. But in this case, this island is close enough that individuals are coming from the mainland. So those individuals are bringing their alleles in. We can see the genes, the individuals come in, and of course they take their genes with them when they come in. So there are two populations of this songbird, the eastern and the central populations. And if you look at this chart on the right, you can see that they have different survival rates. The females in the eastern population survive at a much higher rate than females in the central population. So what actually is happening here is that the central population has a high number of individuals coming in from the mainland, whereas the eastern population does not have as high as such a high gene flow, less individuals coming in. So if you are a female born in the central population, either you are a bird that's already here, or you're a bird that has come in from the mainland and laid your eggs. If you're born in the central population, you have a smaller chance of survival than a female that's born in the eastern population. And they believe that what's going on here is that those birds in the eastern population have had a chance to adapt to their environment. The populations have changed to fit the environment. But the central population, because there are constantly birds coming in who are not adapted to this island, they don't survive as well. And so in this case, the gene flow from the mainland is actually working against that central population because individuals are not surviving as well in that population.